The Tubulin Code. The tube nobody can photograph. A microtubule is a hollow tube. The name gives itself away, spoken slowly, a tiny tube. It is one of the standard building materials of life, found in every cell that keeps a nucleus, which is to say, in every cell of every animal, every plant, every fungus on Earth. Under the microscope, it is a filament, long, straight, and stiff, about 25 nanometers across. 1,000 of them, laid side by side, would span a human hair. The wall of the tube is built from a single repeating stone. The stone is called a dimer, a pair, because it is two proteins locked together, one called alpha-tubulin, one called beta-tubulin. Every stone in the wall is the same pair, the same way up. Dimers stack into straight columns. 13 columns stand side by side, and the columns close into a cylinder. The finished wall, all those stones in their regular array, has a name of its own. It is called the lattice. A lattice of identical stones, 13 columns around, closed into a tube. That is the whole architecture, and it fits on a napkin. It is also one of the most studied objects in all of cell biology. Here is what most studied means in weight. Take brain tissue and dissolve out all the protein that will dissolve. Between 1/10 and 1/5 of that protein is tubulin. The thinking organ is, by a measurable fraction, built of this one stone. Tens of thousands of laboratories have purified it, and the great structural methods, X-ray crystallography first, then cryo-electron microscopy, have photographed the lattice down to its atoms. The portraits are magnificent. And in every one of those portraits, something is missing. Each tubulin protein carries at one end a short, loose stretch of amino acids, about a dozen of them, hanging off the body of the protein, like a thread off a spool. It is called the C-terminal tail, the tail at the carbon end. Proteins have two ends, and chemists name them for carbon and for nitrogen. Tubulin's carbon end was never folded into the stone. It dangles. When the tube is assembled, thousands of these tails cover its entire outer surface. The lattice is shaggy with them, from one end of the filament to the other. The tails are present in every real microtubule ever built. They are absent from every portrait of one. Not trimmed off by the preparers, not hidden inside the wall, averaged away. A structural portrait is an average, and that is the fact this whole chapter turns on. The machines that solve molecular structure cannot look at a single molecule. They need many copies, millions of them, crystallized or frozen, and they build the picture from what the copies agree on. A part that sits in the same place in every copy shows up sharp. A part that sits in a different place in every copy does not blur. It disappears. There is an older way to see the same thing, and it belongs to ordinary photography. Make a long exposure of a busy square at dusk. The building stands still for the whole exposure, and the building is in the picture. The crowd crosses the square, each person in a different place each second, and the crowd is not in the picture at all, not smeared, gone. The structural instruments are that camera. The lattice is the building. The tails are the crowd. The tail holds no pose. Chemists call it disordered, and the word means exactly what it says. The tail waves, it samples shapes, it never settles into one form long enough to be counted. Each copy of the molecule waves its tail differently, and the average of 1,000,000 different positions is no position at all. And the motion is not a private habit of the tail. It is the condition of the world the tail lives in. At that scale, there is no stillness anywhere for anything. Every molecule in the cell is battered from all sides by water molecules, always moving at great speed and getting anywhere slowly. Nothing down there is calm. There is only the storm and the things that have learned to stand in it. Nor is the storm made of water alone. The inside of a cell is crowded past any ordinary meaning of the word. 1/4 to 1/3 of the space is filled with large molecules, sugars, whole machines, all of them jostling, all of them colliding without pause, billions of times a second. The tails do not wave in open water. They wave in a crowd that never stops hitting them. So the part of the tube that never holds still is the one part no portrait can catch. That is not a failure of the instruments. The instruments reported honestly everything that stands still, and the tail's absence from the picture is itself the first real fact about the tail. It is the moving part. It was always going to be the moving part. For a long time, the missing fringe was treated as untidy decoration, the frayed end of an otherwise well-made rope. That reading has the order of things exactly backwards. The lattice is the part with no identity. One stretch of tube is much like another, stone upon identical stone, nearly the same in a skin cell as in a nerve cell, nearly the same in a human as in a yeast. The body of the stone has been kept almost unchanged across more than 1,000,000,000 years, frozen by its own usefulness. Nothing in the cell is less specific than the part the portraits show. The identity lives on the part the portraits miss. The tail is the one stretch of the stone that evolution left free to vary from species to species, and free in every one of them to be written on after the stone is built. It is the surface where one tube is told from another. Every mark this account will ever name is written on the tail you cannot see. In 3 sentences, then, a microtubule is a hollow tube whose wall, the lattice, is 13 columns of a single repeating stone, the dimer, 1 alpha and 1 beta locked together. Every stone dangles a loose tail at its carbon end, and the tail never holds still, so the structural portraits, which are averages, leave it out, present in the tube, absent from the picture. And that missing fringe is not decoration. It is the one free surface on an otherwise frozen stone, and the whole of the Tubulin Code is written on the tail you cannot see. The Tubulin Code, the stones that turned out identical. The first chapter said every stone in the wall is the same pair of proteins. That sentence is true, and it is not the whole truth. The whole truth begins in the late 1970s, when new tools let biologists read genes directly, and the genes refused to be simple. Instead of one gene for alpha-tubulin and one for beta, genomes carried whole families. Vertebrates turned out to keep 6 alpha genes and even more beta genes, each one spelling a slightly different stone. Each variant is called an isotype, the same stone from a different mold. The differences between the molds are small, and they cluster pointedly in one place, the tail, the loose end that no portrait can catch.
A discovery like that comes with a dream attached, and this dream was a beautiful one. If the cell keeps many molds, the reasoning went, it must be because each mold has its own job. One isotype for the spindles of dividing cells, another for the long cables of nerve cells, another for the beating cores of cilia. One isotype, one function. The genome as a stonemason's yard, a different stone stacked ready for every structure in the city. It was exactly the kind of idea a field falls in love with, tidy, sensible, and flattering to the genes. Then the experiment was done, and the experiment was unkind. When different isotypes were offered the chance to build together, they built together, indiscriminately. Alpha and beta variants mixed freely into the same lattice, the same tube, the same stretch of wall, with no sorting and no preference worth the name. The finding has a name of its own: copolymerization, the free mixing of different stones into one structure. By 1987, the verdict was plain. The stones were interchangeable. A mason's yard where every stone fits everywhere is not a yard with a plan. The disappointment was real. Years of careful genetic bookkeeping had seemed to point at a purpose for every variant, and the lattice itself shrugged. But the dream did not die everywhere, and the exceptions deserve their names. In the blood there is a small, clear case. The platelet, the cell fragment that stops bleeding, holds its flattened disc shape with a ring of microtubules called the marginal band, a hoop of tube running around its rim. That ring depends on one particular beta isotype. Take that isotype away, and the ring fails, and the platelet is misshapen. So it was never that no stone ever mattered. It was that the law, one isotype, one function, was not a law. And the story has a second act, delivered decades later by the clinic. In human patients, single damaging mutations were found in single tubulin genes, one isotype, one letter wrong, and the consequences were not subtle. Neurodevelopmental disorders, brains built wrong from the start. If the stones were truly interchangeable, the remaining isotypes should have covered the loss. They did not. The test tube had said the stones were interchangeable. The body was keeping its molds distinct anyway. The question the disappointment was supposed to have closed is open again. So the dream died as a law and survived as a suspicion. The many molds are real. Their differences are real, and they sit in exactly the place this account has already marked as the writing surface. The tails. Whatever the isotypes are for, it will be read there, on the loose ends.
And one thing more should be set down now for later. The dream, many stones, many jobs, felt so natural that nobody thought to ask whether nature had ever run the experiment the other way. One stone, many jobs. Nature had. There is an organism that never had the dream, and it will be heard from again. In 3 sentences, then. Genomes carry many tubulin genes, encoding many isotypes, the same stone from different molds, differing mostly in the tail. The dream that each mold had its own job was killed by copolymerization, the free mixing of isotypes into one lattice, but it left survivors, the platelet's marginal band and the human disease mutations that prove one damaged mold cannot always be covered. The stones turned out identical where it was easiest to look, and not quite identical where it mattered most. The Tubulin Code. The clock.
Every tale this account follows ends in the same brick. The very last amino acid of the alpha-tubulin tail is, almost always, a tyrosine. The word is old and homely. In 1846, Justus von Liebig isolated the stuff from cheese protein and named it from the Greek word for cheese. The most studied protein in the brain ends, of all things, in a piece of dairy nomenclature. Two enzymes spend their whole careers on that one brick. One clips it off, the other pastes it back on. The clipping has a name, detyrosination, the taking off of the tail's last letter. The pasting has a name too, tyrosination, the putting back. And the paster has a name of its own, TTL, tubulin tyrosine ligase, the enzyme whose entire job is to glue a tyrosine onto a tail that has lost one. The clipper, for reasons this chapter will make plain, went for most of its history by a description rather than a name, the D-tyrosinase, the enzyme that takes the tyrosine off. One brick, two hands, an on and an off. It sounds like the smallest circuit in the cell. It is a clock. Here is where the story stops being ordinary chemistry, and it is worth hearing slowly, because the whole clock is in it. The two enzymes do not work on the same material. The paster works only on loose stones. TTL can reach a dimer floating free in the crowd, a stone not yet built into anything, and it cannot work on a stone that is standing in a wall. Its grip is shaped for the loose form. The built lattice offers it no purchase. The clipper has the opposite taste. The D-tyrosinase works far better on tubulin that is already assembled, already standing in a microtubule. Loose stones barely interest it. The built tube is its territory. Two enzymes, two tastes, and the tastes never overlap. They do not compete over the same molecule, ever. They work in different territories, divided by a single fact, whether the stone is in the wall or out of it. This is sub-story enough on its own, and the field gave it no ceremony when it was found. It was measured, published, and filed. It deserves a slower reading, because everything the clock does follows from it. Now follow what the division of labor does to one filament over its life. A fresh microtubule is built from loose stones, and loose stones are the paster's territory. So the stones arrive fully capped, every tail ending in its tyrosine. The moment the wall stands, it crosses a border. It enters the clipper's territory, and the paster can no longer touch it. So the standing tube is clipped, tail by tail, at the clipper's working pace. The pace has been measured. To mark a standing microtubule completely takes several hours, and the two great regimes of cell life show the result plainly. In a skin cell, where microtubules turn over quickly and fall apart young, few filaments ever carry the mark. They do not stand long enough to earn it. In a nerve cell, where microtubules stand for long stretches, most of them do. The clipped mark is, by mechanism, the property of old tubes. The tubes that stand longest are called the stable, long-lived microtubules, and long-lived is the honest word, because what makes them survive is a story of its own, and it arrives 2 chapters from now.
One more turn, and the clock is complete. The mark lasts exactly as long as the wall. When a clipped microtubule finally falls apart, and in the life of these filaments, everything falls apart eventually,
its stones drop back into the loose pool, and the loose pool is the paster's territory. TTL meets them there and pastes the tyrosine back. The brick that was taken off the standing wall is restored to the fallen stone, and the stone goes back into circulation, young again on paper. Age is written only while the structure stands, and it is wiped clean the moment the structure falls. Step back and look at what has been built here. No enzyme measures time. No protein carries a clock. There is no timer anywhere in this machinery, only which enzyme can reach which stones. And yet the cell's oldest filaments are chemically recognizable as old. Time itself has been converted into a chemical state by two enzymes with opposite tastes in scaffolding. The clipper in this story has a biography, and it is the strangest search in the field. It begins in the mid-1970s in Córdoba, Argentina, in the laboratory of Ranuel Caputo, where Carlos Arce and Héctor Barra showed that brain extracts could perform both halves of the cycle, remove the tail's last brick, and restore it. The cycle was real. Both enzymes existed. What remained was to find them. The paster was caught within a few years. TTL was identified, purified, and studied, and it has been a known enzyme ever since. The clipper refused. Everyone knew it was there. The clipped tails were there to prove it, accumulating on old tubes in every cell anyone examined. The activity could be measured in a test tube. Add the extract, watch the tyrosines come off. But an activity is not a molecule, and no laboratory could corner the molecule responsible. Not in the 70s, not in the 80s, not in the 90s. Generations of researchers retired from the hunt. The D-tyrosinase became the field's famous missing person, an enzyme whose work was everywhere and whose face was nowhere. A great review written in 2014 could still only call it the unidentified D-tyrosinase. 40 years went by like that. When the answer finally came in 2017, it came the way these answers sometimes come, from two laboratories at once, independently, publishing side by side in the same journal. And the answer carried a twist that 40 years of failure had somehow earned. The missing clipper was not missing. It had been catalogued since 2004 under a name from a different country of science, vasohybin, a protein first found in the cells lining blood vessels and studied for more than a decade as a brake on blood vessel growth. The enzyme that writes the age of microtubules had been sitting in the literature the whole time, with a name, with a job description, filed under blood. Consider what that means plainly. For over a decade, two files were open in two rooms. In one room, a nameless enzyme that clipped old microtubules, its work measured and remeasured, its identity a standing embarrassment. In the other room, a named protein that slowed blood vessels, well-studied, well-funded, its deeper chemistry unexamined. Nothing in either file pointed at the other. Nobody had a reason to carry one folder across the corridor, until two laboratories in the same year carried it. The case was never cold, it was misfiled.
There is a lesson in the 40 years, and it is not a sentimental one. The cell does not organize its chemistry the way libraries organize knowledge. An enzyme is one molecule with one active site, and it will do its job in whatever context the cell puts it. Blood vessels on one day of the week, microtubules on another. The file system is a human overlay. The chemistry was one chemistry all along. So the cell's oldest roads are chemically recognizable as old. The mark is not an order. Nothing commands anything. It is evidence of time past, left on a surface that other proteins can feel. A clipped tail on a standing tube is like a gray hair. To say it once and put the image down. The metaphor is retired here. What remains is the mechanism. 2 enzymes, 2 territories, and age written only while the structure stands. In 3 sentences, then, the alpha tail ends in a tyrosine. TTL pastes that brick onto loose stones only, and the D-tyrosinase clips it preferentially from built tubes. And that division of labor, all by itself, turns a filament's age into its chemistry. The clipper hid for 40 years and was found in 2017 in 2 laboratories at once, already catalogued under the name vasohebin, filed under blood. And the mark lasts exactly as long as the wall it is written on, which makes the oldest roads in the cell the most legible ones. The Tubulin Code, the permanent mark. The clipping can go further, and the further clipping changes everything. The alpha tail ends in tyrosine, that was the last chapter, but the brick just before the tyrosine is a glutamate, and enzymes can remove that brick too. The first loss is reversible. While the glutamate stands, the paster can always put a tyrosine back. The second loss is not. A tail missing both bricks, the tyrosine and the glutamate beneath it, has crossed into a state no enzyme can undo. It has a name, delta-2 tubulin, 2 letters lost from the end and gone for good. The reason is a small, strict piece of chemistry, and it is worth holding. The pasting enzyme needs the glutamate. TTL does not glue a tyrosine onto a bare tail. It anchors on that penultimate glutamate, the way a hook needs its peg. While the peg stands, the door is open. Tyrosine off, tyrosine on, as often as the cell likes. Take the peg, and there is nowhere left to anchor. The door does not swing shut, it locks. So the cell keeps 2 marks of age, one soft and one hard. The soft mark is the clipped tyrosine, a mark that lasts exactly as long as the wall, wiped clean the moment the stone falls back into the loose pool. The hard mark is delta-2, a record of age that no repair enzyme can touch, carried by the stone for as long as the stone exists. And the clipping does not necessarily stop there. Tails have been found missing more than the last 2 bricks, a state called delta-3, and what delta-3 means nobody knows. It sits in the literature like a locked room off a locked corridor. Now the number that makes this matter. When biochemists measured brain tubulin directly, they found that about a third of it, roughly 35%, is delta-2. A third of the brain's microtubule protein is in the state that can never go back. This is not a rare scar on a few veteran filaments. It is a mass condition of the organ. The brain of all organs runs on microtubules that have crossed a point of no return. And here the account must be honest, because the field is honest. Nobody yet knows the full meaning of that number. The mechanism is known. One more clip, and the reversible mark of age becomes an irreversible one. The purpose is not known. The open file stays open in this film.
What happens when the lock is tested at the scale of a whole animal is known, and it is grim. Mice born without the pasting enzyme, no TTL at all, accumulate D-tyrosinated and delta-2 tubulin across the board, and they die before birth, their nervous systems built wrong. A cell that cannot recap its stones drowns in permanent age. Who takes the glutamate? These erasers are known, and they are a family. The cytosolic carboxypeptidases, the CCP family, enzymes that work in the cell's fluid and trim amino acids from the ends of tails. The same family cuts into the gene-encoded tail itself to make delta-2 and delta-3. One family of erasers, working both on the marks and on the tail beneath the marks. Hold three facts together. A third of the brain's supply is locked. The lock is one missing brick, and the key was thrown away by an enzyme doing its ordinary work. There is no drama in the mechanism, and that is exactly the point. Permanence in this cell is not a special event. It is what happens when one ordinary erasure meets one strict requirement of one repair enzyme.
And these erasers are not finished with this account. The CCP family, the erasers from the locked door chapter, will return in the darkest chapter of the film, in a role nobody would wish on them. In 3 sentences, then, beneath the tail's last tyrosine sits a glutamate, and removing it makes delta-2, a mark that can never be repaired, because the pasting enzyme needed that glutamate as its anchor. The erasers are the CCP family, which also cut further to delta-3, whose meaning is unknown. And roughly a third of the brain's tubulin carries the locked mark, a mass permanence at the center of the organ, whose meaning remains, honestly, an open file. The Tubulin Code. Why Old Tubes Survive. The last 2 chapters made the old tubes recognizable. They stand, they are clipped, and some of them are locked past repair. One question was left standing with them, on purpose. Why do they survive at all? The obvious answer was wrong, and it was believed for years. Old tubes carry the clipped mark. Old tubes survive. Therefore, the reasoning went, the mark must stabilize the tube, stiffen it, brace it, make the lattice itself harder to tear down. It is the kind of inference that feels like common sense. It is not what the mark does. The clipped tail changes nothing about the strength of the lattice. The tube is not reinforced. Something else is happening, and it is more interesting. The cell keeps enzymes whose profession is taking microtubules apart. They are called the kinesin 13s, and 2 of them have names worth knowing, KIF2 and MCAK. They belong to the kinesin motor family, but they carry no cargo. They are depolymerizers. They find the end of the tube and pry it down, stone by stone. The demolition crew always on shift. Given free run, they are the reason no microtubule would stand for long. Here is what the clipped mark actually does. It wards the demolition crew off. Kinesin 13s work poorly on D-tyrosinated tubes. The clipped tails turn them away from the wall they came to dismantle. The mark does not make the wall stronger. It makes the wreckers weaker where they grip. Old tubes survive not because they are braced, but because they are guarded.
The clipped tails stand along the road like bodyguards, turning the demolition crew away at the curb. And the reading runs in both directions, which is the quiet marvel of this chapter. The absence of the tyrosine is read by the wreckers. The presence of the tyrosine is read too. There is a small protein domain, cap-gly, carried by proteins called CLIP170 and P150 glued, that binds the tyrosinated tail specifically, the capped tail, the fresh one. Proteins with this grip ride the growing ends of young microtubules like scouts posted at the front of the advance. So the last brick of the tail is never neutral. Capped, it is read by the riders of young tubes. Clipped, it is read by the crews that spare old ones. One letter at the end of the tail, and both of its states have readers. So the sentence to keep is a corrected one. Detyrosination does not stabilize the tube, it protects the tube by refusing the enzymes whose trade is taking tubes apart. And the unclipped state is not a blank state, because the tyrosine itself has readers of its own. Presence is read and absence is read, and between the two readings the cell sorts its roads by age without a clock anywhere. Stable was the wrong word all along. The old tubes are not stronger, they are better defended. And a road that is defended rather than braced can still be taken down the moment the guards are called off, which is exactly the kind of road a living cell needs. Permanence was never on offer. What was on offer was a truce, posted in chemistry, readable from both directions, and revocable at will. The marks so far were removals, a clipped brick at the tail's end, taken off and sometimes put back on. The cell's writing had been done with an eraser, and the writing was real. But nothing had been added yet, nothing built. Now the account turns to the first mark that adds, the first mark that constructs something on the tail, out of the tail's own material. The material is glutamate. The name comes from glue. A chemist named Ritthausen pulled the acid out of wheat gluten in 1866 and named it for what it came from. The tail is rich in glutamates, and the cell keeps enzymes that can take any one of them and use it as a hook. From the hook, a chain grows, glutamate after glutamate, strung one behind the next, hanging off the tail like beads off a thread. This is polyglutamylation, the stringing of acid beads on the tail. It was found in 1990 on tubulin taken from the brain, and brain tubulin carries it heavily. It has been found heavily everywhere the cell keeps its most demanding structures, the bundled cores of cilia, the bodies that organize the lattice. But the brain is where it was first seen, and the brain is where it is densest. The bond that holds the chain deserves one sentence because it decides the shape. In an ordinary protein, each amino acid joins the next end to end in a single unbroken line. Glutamate carries a second acid group out on its side, a spare socket. The first bead of the chain plugs into that spare socket, not into the line. The chemists call it an isopeptide bond, a bond that steps sideways. So the chain does not lengthen the tail, it branches off it, a string of beads growing out into the water, off to the side of everything, hanging free. Now the finished picture is worth holding whole, because it will be said three ways, and all three are the same fact. Said as chemistry, the cell hangs a chain of acid beads on the tail. Said as physics, each bead is a charge. Glutamate is an acid, and an acid in water sits there as a small negative point. The tail is charged already, its charge fixed at birth, written into the gene. Every bead strung on the chain adds one more negative point. A heavily beaded tail is not merely longer, it is more densely, more strongly negative than a bare one. Said as physics, the cell raises the charge of the surface. And said as counterfactual, said by looking at what could not happen otherwise, charge is the one thing a reader can feel. The proteins that walk and work on the wall touch it through charge alone, attraction and repulsion, nothing else. An unmarked tail has one fixed feel, set the day the gene was written, and nothing downstream can change it. Evolution can rewrite the gene over generations. Nothing inside a living cell can retune a bare tail. The chain is the only tool the cell has for changing the feel of a tail after the stone is built. That is what the mark is for. Not a message, a change in what the surface lets happen next. The chains do not string themselves, and the family that strings them earns a telling of its own. The enzymes go by a dry name, TTLL, and the family was identified only in 2009, which means the writers of the best-known mark in the brain were nameless for 2 decades after their work was found. A mammal carries 9 of them, 9 specialists.
The specialization is a division of labor, and it runs in 2 directions at once. First direction. Some of the 9 can only start chains. They find a bare glutamate on a tail, hang the first bead, and their trade is finished. Others cannot start anything. They only extend. Give them a chain already begun, and they add bead after bead after bead. Initiation and elongation, the starting and the lengthening, are different trades, and no single member of the family practices both well. Second direction. The specialists have opinions about which tail they touch. Every stone in the wall is 2 proteins locked together, an alpha and a beta, and each carries its own tail into the water. Some of the 9 work the alpha tail, some work the beta. The preferences are built into the enzymes themselves, not instructed from outside. It is worth sitting with what this means. The length of a chain on a given tail, 3 beads, 8 beads, is not weather. It is the outcome of which specialists were active there, starters against extenders, working which tail. The number of beads on a tail is a number the cell set, through a committee that never meets. That number is worth holding. The next chapter is about what it controls. A mark is not a message. It changes what may happen next. The acid beads are the purest case yet. A chain of charges, hung bead by bead, its length set by a committee of 9, felt by everything that walks the surface. The road's feel is charge, and the chain is how the cell changes the feel of a road it has already built. So the wall stands as it always stood, patient, stone by stone, but its surface has changed under the account's hand. Some of its tails now hang heavy, branched, beaded, dense with negative charge, each chain the length a specialist left it. The beads wave in the same water the tails wave in, reachable from every side by any enzyme that swims close enough. From a distance, nothing. Up close, a rewritten surface. A rewritten surface will be read. The beads are about to set the speed of a machine, and after that they are going to overgrow, and a mouse is going to pay for it. The chain of charges is the account's center now. Everything that follows,
hangs from it.
Some machines on the lattice do not read the road to walk it, they read it to cut it. Spastin is the name to hold, a severing enzyme, a molecular pair of shears. It grips the filament, threads one of the tails through itself, and cuts the tube in two. Cells use it to prune their networks, to remodel them, to demolish a road on demand and build a better one over the rubble. In neurons, it trims unwanted branches, and its loss is not abstract. When the human spastin gene is broken, the longest cables of the spinal cord degenerate, and the legs stiffen year by year. The shears matter, and the tails are their handle. Spastin takes hold of the wall by the writing on it, which raises the question this short chapter exists to answer. The shears work on the writing. What sets their speed? The acid beads from the chains chapter. Now watch what they do. In 2010, a team led by Benjamin Lacroix and Karsten Janke measured the cutting. The more beads a stretch of lattice carried, the more strongly the shears were drawn to it, and the faster they cut. The mark does not tell the machine anything. It changes how fast the machine works. Then the picture sharpened into something stranger and more precise. Later work, Wallenstein and Roll-Mikak, 2016, built tails with chains of known length, timed the cutting, filament by filament, and found that the relationship is not a switch, it is a dial. Cutting rises as the chain lengthens, up to an optimum, a best length, and past that length, more beads slow the cutting back down. Too few beads, and the shears barely work. Too many, and they slow again. Somewhere between, a speed the cell can aim at. The field's name for this shape is biphasic, rising, then falling. The spoken name is simpler, a dial with a sweet spot. Why the cutting falls past the optimum is still argued over. The honest state of the record is that the rising arm is measured, the falling arm is measured, and the cause of the fall is not yet settled. The dial stands either way. Remember what a mark was claimed to be, not a message, but a change in what may happen next. Here, that claim stops being philosophy and becomes measurement. The number of beads on a tail sets the rate at which the tube will be cut, a physical quantity on a surface setting the speed of a machine. That is the code in its least metaphorical form. 3 things to carry out of this short room. The shears read the beads. The bead count sets the cutting speed, rising to an optimum, falling past it. And a mark, for the first time in this account, has been caught doing its work as a number, not a word, not a letter, but a quantity a machine obeys. One shadow falls forward, and it is worth naming before it arrives. A dial that rises and falls implies a hand that keeps it in range. If nothing trims the beads, if the chains grow and nothing answers, the dial runs off its scale into the region where the reading goes wrong. The next chapter is the record of exactly that failure. It is the darkest page in this account. Every hand that writes is answered in this account by a hand that erases. The writers string the beads. Somewhere an eraser trims them back. Neither hand ever rests. The state of the lattice at any moment is the balance between the two. The code was never the marks.
The code is the balance between the two hands. The erasers from the locked door chapter, the carboxypeptidases, the enzymes that clip marks back down, return now. In that chapter, they made the permanent mark, the one no repair enzyme can undo. Here, they return in a darker role. This chapter is about what happens when one of them is lost, and it is told at the tempo the record was measured at, slowly, one step at a time, without looking away. The chains again, the same acid beads from the chains chapter, the same branched strings of negative charge. A young neuron, growing its long cable, beads its tubulin heavily as it matures. Becoming a neuron means, in part, accumulating these marks. So the brain keeps a dedicated eraser for them, an enzyme called CCP1, the brain's main D-glutamylase, its trimmer of bead chains. Wherever a chain has grown too long, CCP1 is the hand that shortens it. Now ask the hard question plainly. What happens if the hand is gone? The answer has a name: hyperglutamylation, overbeading. Chains that should have been trimmed keep growing. Tails that should carry a few beads carry many. The surface charge climbs past anything the machinery evolved to feel. The marks pile up until they mean nothing, the way a page full of underlining underlines nothing. And remember what the dial chapter measured. Past the optimum, the shears slow, and the reading of the surface changes character entirely. Overbeading does not mean more of a good signal. It means a signal the machinery can no longer make out. The answer also has a body, a small mouse called PCD, Purkinje cell degeneration, and its record is told whole, flatly, because the record is what the sentence costs. The Purkinje cells are among the longest, most transport-dependent cells in the brain. Their far reaches must be supplied, every twig, by motor traffic on microtubule roads. In the PCD mouse, these cells are born healthy, and then they die young. The decline is quick by the clock of a laboratory. Within weeks of birth, the cells begin to fail, and the mouse does not live out a normal span. For a long time, the cause was a mystery wearing a mask. Then, in 2010, the lesion was found, and it was not in a writer, it was in an eraser. The mouse's CCP1 was broken. One eraser, gone. And here is what followed, step by step, each step measured. With nobody erasing, the bead chains overgrew. The roads became overmarked. Motors that should have walked those roads hesitated. Cargo stalled. The far reaches went hungry. And the longest, most transport-dependent cells in the brain died first. Not because anything attacked them, but because a balance failed. The field checked the slope from the other direction to be sure of it. Mice were engineered to overbead their tubulin directly, eraser intact, writers pushed. The result was the same disease, disturbed axonal traffic, dying neurons. Excess, by itself, was enough to kill.
Then the record turned, and the turn is the reason this chapter exists. If the disease is an unopposed writer, what happens if the writer is weakened? The experimenters removed TLL1, one of the 9 specialists, the one elongating chains in these cells, from the dying mouse. They fixed nothing. The eraser stayed broken, but with the writer's hand slowed, the chains stopped overgrowing, the traffic recovered,
and the Purkinje cells survived, not for a week, not for a reprieve, but into the mouse's old age. Balance, restored from the other side, held. The human parallel exists, and it is stated once without ornament. Children with failures in the same eraser develop infantile-onset neurodegeneration, the same lesion, the same slope. Writers need erasers. The mouse is what the sentence costs when it is not true. Enough is an address, too much is a disease. The cell does not keep erasers for tidiness. It keeps them because an overmarked road is as broken as a missing road, and the price of a broken address system is paid, first, by the cells that depend on it most. There is one more thing in the rescue, and it deserves the last quiet beat. The cure was not repair. Nothing was fixed. A second defect was introduced to cancel the first, a weakened writer balancing a broken eraser, and the cells lived. That is what a balance means, not the health of either hand, but the tension between them. The death of the Purkinje cells has an old shape, the oldest in this account. Growth is patient, catastrophe is sudden. The cells were there day after day, and then they were not. A balance kept, a balance lost. And the loss, when it came, did not come gradually.
Every mark so far has been a generalist. The clipped brick, the acid beads, they turn up on roads all over the cell. Now the account comes to a mark with one habitat, and one habitat only, a specialist's mark. The habitat is the cilium, the beating hair of a cell, the slender finger some cells wave to move fluid or hold still to sense. And the mark is polyglycylation, the stringing of glycine beads. Glycine, the smallest amino acid there is, a bead with almost no side to it, a bare pebble. Same architecture as the glutamate chains, the same sidewise isopeptide bond, the same branching strings, different bead. The chemistry will sound familiar, and it should. The cell hangs a chain of glycines on a tail glutamate, bead after bead. Monoglycylation is the name for the first state, one bead, the branch point alone. Every long chain begins as one bead. Many tails never go further. The single bead and the strung chain are different marks, and the cell keeps them separately. The writers are more of the 9 specialists. TTLL3 starts the chains. TTLL8 and TTLL10 extend them. The division of labor, again, starters and extenders, now laid on a different bead. And the habitat is exact. The axoneme concentrates on the mark. The axoneme is the most heavily marked structure in this whole account, beaded with glutamates, beaded with glycines, clipped at its tail ends. Some cilia even pick the glycine mark up as they mature. A young hare carries the single bead, and the long chains come later with age. If the code has a capital city, this is it. Now the lost enzyme and the question it leaves behind. Human TTLL10 is dead. The gene is still in the genome. Anyone can find it, spelled out, intact enough to read. But the enzyme it encodes does nothing. Somewhere in the human lineage, one of the chain extenders stopped working, and the lineage kept the gene anyway.
the way a house keeps a key to a lock that no longer exists. The consequence is plain. Humans cannot elongate glycine chains. Other animals string long glycine chains on their axonemes. Ours stay short, a bead or two, the first state and little more. And here is the uncomfortable fact, stated flatly. We get by. Our cilia beat. The fluid in our airways moves. The sensing hairs sense. Long glycine chains, which other creatures spend working enzymes to build, are a thing the human body does without. So what is elongation for? The honest answer is that the record does not say. An honest gap stays honest. But the record offers one more measurement and it complicates the shrug. Take the glycylation away, deplete it, in the ciliated cells that do string these chains, and the axonemes do something very specific. They build, and then they fall apart. Assembly succeeds, maintenance fails. The hair goes up on schedule, complete, and then it cannot keep itself. That is a strange finding, and it is worth one still moment. The mark is not needed to build the structure. It is needed to keep it. A road can be laid without the beads. It cannot be held without them. Not essential until it is. Most marks in most places can be lost one at a time, and the cell carries on. The cilia are where the losses show. The glycine chains are the clearest case in the account, a mark the body can do without nearly everywhere, except in the one structure that cannot do without it.
So the map of marks now has a coastline. Some writing covers the whole country, the clipped brick, the acid beads. Some is confined to one small territory, the beating hairs, and in that territory it is load-bearing. And the human line has let one of its writers die and walked on, chains unlengthened, asking without asking, what was the lengthening for? The next chapter goes to the one place such questions can be put cleanly, a small creature in pond water with exactly one kind of stone that answers with its life. A doubt has been standing in the corner of this account since the stones chapter, and it is time to face it. The stones come in different models. The genes give slightly different tubulins, differing mostly in their tails. And the marks differ. Two sources of difference stacked on the same wall. So when a cell gives one microtubule a special job, which is doing the specifying, the model of the stone or the writing on it? In a creature with many models, the two can never be fully separated. Every experiment inherits the confusion. Nature has already run the control experiment. It is a creature from pond water, and it is the organism that never had the dream, the isotype dream, one model for one job, the dream the cloning era buried. This creature never had the dream to lose because it only ever had one stone. The creature is tetrahymena, a single cell covered in beating hairs, small enough to swim a drop of pond water and old enough to have seen most of evolution. Its entire genome holds exactly one gene for alpha-tubulin and one gene for beta-tubulin, one model of stone, no variety at all. Every microtubule it builds from the same two proteins. And yet, this creature runs one of the richest mark repertoires known. It strings glutamate beads, it strings glycine beads,
It clips its tails. Different marks on the hairs, different marks on the mouth parts, different marks on the division machinery, each structure written differently, each matched to a different job. Whatever specialization means in a microtubule, tetrahymena achieves it through the marks alone. One stone, many roads, and the roads told apart entirely by their writing. There is a second gift in this creature, and it is the one the experiments run on. Because its tubulin is one gene, experimenters can rewrite that gene directly, rewrite the tail, mutation by mutation, and watch what changes. No shelf of isotypes to confuse the answer. The marks can be studied against a perfectly uniform background in a living cell. 3 experiments, and they climb. First, the usual modification sites, the tail glutamates that normally take the beads, were removed, one mutation at a time. The expectation was damage. What happened instead? The enzymes picked other sites. The writers did not know the map had changed. They write where they can reach, and a reachable glutamate is an address. Beads appeared on glutamates that are never used in a normal creature, in the wrong places, and the creature swam on. The writing went to the wrong addresses, and the wrong addresses sufficed. Second, all of them, every usable site stripped from the tail, the surface left with nothing to write on, and this the creature could not survive. It died. Not because any single mark was missing, because the capacity to be marked was gone. The tail was still there, still waving in the water. It simply could no longer be written on, and that was fatal. Third, the chimera. Experimenters cut the tail off the beta stone and grafted it onto the alpha, so that both tails on every stone were beta tails, and the alpha positions became wrongly, heavily overglycolated, marked in places no alpha tail is ever marked. A miswritten creature by any exact standard. Nothing in the stone itself objected. It lived. It swam. Excess writing in the wrong place on the wrong stone tolerated. Hold the 3 results together because they are the strongest evidence this account will offer. Wrong sites, tolerated. Wrong tail, wrong stone, too much writing, tolerated. No sites at all.
The cell does not need the writing to be exact. It needs the writing to be there. No single mark in its right place is the point. The point is a marked surface, a climate of charge the machinery can live in within wide margins. The creature dies not when the message is wrong, but when there is nothing left to write on. That is the sentence the 12th chapter will stand on. The marks work within a range, not at a point, and a code that works within a range is a different kind of code than the word suggests. So the old doubt has its answer from a creature in a drop of pond water. Is it the models or the marks? Tetrahymena has no models, and it specializes everything. The marks suffice. One stone, a huge repertoire of marks, a life built out of the difference between them. The lattice is marked, and the marks are red, and the marks, it turns out, are enough. Every mark in this account so far has had an address, and the address has always been the same, the tails. The clip lives there. The permanent clip lives there. The chains of charge hang there. All of it on the outside of the tube, out where the traffic walks.
One mark breaks the geography. It is called acetylation, the K40 acetylation of alpha-tubulin, and even its name comes from somewhere else, from acetum, Latin for vinegar, the root that named acetic acid. The chemistry is the same one that sours wine, and the mark is written on the inside. A microtubule is a pipe about 25 nanometers wide, and it is sealed along its length. Its inner surface, the lumen, the hollow, is the quietest place in the cell's skeleton. No tail reaches it, no motor walks it, nothing touches it at all. Deep in the body of the alpha stone there is a lysine, one amino acid among hundreds at position 40. K40, the field calls it. When the stone is built into the wall, that lysine ends up facing the hollow. An enzyme can fasten an acetyl group there, the vinegar mark. And of all the known marks on tubulin, this is the only one on the inside. What the inside mark keeps company with is age. It gathers on microtubules that have stood a long time, the long-lived tubes, the same ones that carry the clip on their tails outside. Said carefully, the mark does not make a tube long-lived. It reports that the tube is. The writer of the inside mark carries two names, alpha tat 1 in one literature, mech 17 in another, and one remarkable property. It is slow. A slow enzyme working in a sealed pipe. That is the whole mechanism, and it deserves to be taken apart. A tube that lives for a minute gives the writer no time. Visited perhaps once, it falls back into the crowd of loose stones nearly unmarked. A tube that stands for an hour is visited again and again, and the marks accumulate. Nothing counts, nothing measures time. The slowness of the writer and the shelter of the pipe together do the whole job, a mark that reports age built without any clock. This is the second age signature in this account, and the two are worth setting side by side. The clip outside is cut by an eraser that prefers built tubes. The vinegar mark inside is laid by a writer that works too slowly to bother with young ones. Two different enzymes, two different surfaces, one result. The oldest roads in the cell are the most legible. And yet the two signatures are not the same kind of fact. One sits on the outside, where the readers walk, and readers have been caught reading it. The other sits in the hollow, where nothing walks at all. Why a mark of age should be written in a place no reader can reach is one of the genuinely open questions in this field, and it will stay open a moment longer because it belongs to the honest part of the record. And here the geography turns back on the beginning because one sentence from the first chapter has been waiting for this mark. The tail you cannot see, the tail no structure has ever photographed, the part of the stone where the cell was said to write. Every mark this account has named hangs on that tail. This one hangs nowhere near it. The one mark written where no camera looks is also the one mark not written on the tail that no camera can catch.
The rest of the record is honest, and it stays honest. How the writer gets into the sealed pipe is unknown. The field has not finished answering it. What the mark does is half known. In living cells, acetylated tubes seem to route motor traffic differently. In a glass dish with clean components, the effect does not reproduce.
Something is missing from the dish, and nobody has named it yet. The readers, if there are readers, are thought to be proteins sitting inside the lumen, and those proteins are barely characterized at all. The mouse without the writer has been built. It carries no vinegar mark anywhere, and it is nearly normal. Two things falter: the sperm flagellum and a small civility of crowded cells called contact inhibition, the rule by which cells that touch one another stop dividing. That is the whole phenotype of the missing inside mark. It has erasers, two of them, HDAC6 and CERT2, and neither is faithful to this one job. Both erase elsewhere, too. So the early experiments that blocked them must be read with care. Whatever was measured was never the vinegar mark alone. In 3 sentences, then, one mark, acetylation, the vinegar mark, is written not on the tails, but on a lysine facing the hollow of the tube, the only mark on the inside. Its writer is a slow enzyme in a sealed pipe, and that slowness, in that shelter, lets the mark accumulate only on tubes that have stood a long time, a report of age with no clock anywhere. And how the writer gets in and what the mark is for remain open questions, the field's own and the honest kind.
In 2007, the field gave the whole arrangement a name. Kristen Verhey and Jacek Gerteg called it the Tubulin Code, and they borrowed the word deliberately. 6 years earlier, researchers studying how cells package DNA had spoken of a code of marks on the packaging proteins, a histone code, and the word traveled from the packaging people to the microtubule people. Code is an older word than any of this. It comes from codex, the Latin for a block of wood, which became the word for a book, a thing one writes on, and then a thing one reads. The borrowing was elegant. The question of this chapter is whether it was true. It is now possible to say precisely where the word fits. The marks are combinatorial. A tail can be clipped or capped, bare or beaded, long-chained or short, and the combinations carry different consequences, different motors preferring, different shears tuned, different addresses written. The marks are written by one set of enzymes, read by proteins with preferences, erased by another set. That much is genuinely code-like. And it is possible to say precisely where the word misleads. Nothing decodes. No machinery reads the marks the way these words are being read, extracting an instruction and executing it. The mark on a tail is a physical quantity, a charge, a handle, an anchor point, and what it does is set rates and preferences. It changes what may happen next. The borrowed word deserves a fair trial, though, because the original code, the histone code, is stronger than the tubulin one in exactly the respect where the tubulin one is strange. That contrast is the heart of the matter, and it earns a full telling. The histone code works close to the literal sense of the word. A precise pattern of marks on a packaging protein, this mark here, that one beside it, is bound by a precise reader whose whole shape is built to recognize that pattern and no other, and the outcome follows specifically. This gene opened, that one kept shut. Pattern, reader,
consequence, all matched like wording to meaning. It is a telegram, exact words carried exactly, answered exactly. The tubulin marks send no telegrams. Each mark biases a rate, tilts a preference, nudges a probability. Similar marks give similar readouts. The field's own phrase for this is probabilistic signaling. The signal shifts the odds, and it does not issue the order. A climate, not a telegram. A climate makes some weather more likely. It schedules no storms. This is not a defect being excused. It is the finding, and it rests on evidence that has been accumulating, chapter by chapter, throughout this account. 3 landmarks are already standing, and now they can be fired in one volley, because each of them, seen again, is the same shape, a signal that works within a range. The dial, the rheostat of chain length. The cutting enzyme, spastin, reads the chains of charge, and its speed rises with the number of beads on a tail, up to an optimum, and past the optimum, down again. Not a switch that says cut, a dial with a best setting, answered across a range. The pond creature, the proof by tolerance. Tetrahema, 1 gene for each kind of stone, a huge repertoire of marks. Rewrite its 1 gene and kill the usual modification sites, and the enzymes simply choose others. Kill every site, and the creature dies. And strangest of all, graft a beta tail onto an alpha stone, and the grafted tail is overmarked, loaded with more glycine chains than any natural tail carries, and the creature lives. The system does not proofread the letter of the signal. It tolerates until the signal is gone entirely.
The 9 specialists. The redundancy of the writers. 9 enzymes write the chains of charge, with overlapping preferences. Some favor the alpha stone, some the beta. Some start chains, some extend them. Remove any one hand, and the writing mostly continues. The code is written by a committee, and the committee survives its members. A dial, not a switch. Tolerance, not proofreading. Redundancy, not precision. 3 experiments, 3 laboratories, 3 different marks, 1 shape. And the shape has a name already given. Redundancy and tolerance, the properties a signal has when no single letter of it carries the whole meaning. And the shape explains itself. A lattice that lives on the edge of catastrophe cannot afford a code of brittle exactness. A tube can vanish in seconds by design. A signaling system written on such a structure has to be one that bends and still means. Signals that survive imprecision, that sum and average and hold their sense across a crowd of slightly different tubes. Probabilistic signaling is not the Tubulin code falling short of the histone code. It is the only kind of code a fountain can carry. So the name audit closes, and the word is kept, knowing what it costs. It costs the telegram, no sender, no message, no decoder. It buys the alphabet, combinatorial marks, distinct writers and erasers, readers with preferences, consequences. Code is half the right word, and the half it gets right is the half this account needed. In 3 sentences, then,
The Tubulin Code was borrowed in 2007 from the Histone Code, and the borrowing is exact in one way. The marks are combinatorial and consequential, and misleading in another, because nothing decodes them into instructions. The evidence of the whole account says the marks form a climate, not a telegram. The dial, the pond creature, and the 9 specialists each show signals that bias and tolerate and work within a range. And that is the design, not the flaw. The only kind of code a structure built to vanish can carry.
Every claim in this account was, at some point, measured in a glass dish. The motors walking on marked and unmarked track, the shears cutting faster on beaded tubes, all of it began as chemistry done on purified material, outside any cell. And almost every one of those measurements began with the same material, tubulin, purified from brains. Brain tubulin, chosen for the honest reason that brains are rich in it, and it comes out of them in quantity. There is an irony in that choice, and the field knew it. Brain tubulin is not one thing. It is every isotype the brain expresses, wearing every mark the brain writes, mixed together in one preparation. A third of it carries the permanent clip. The chains of charge were first found on it precisely because it carries them so heavily. A mixture of everything, a soup. Which means that decades of careful measurements measured a soup. Every clean number ever read off an in vitro experiment was the average of a crowd of different stones carrying different marks, all answering the question at once, all blurred into one another. When an experiment reported that a motor prefers old track, no one could ask which stone model, which tail, which combination of marks the preference belonged to. The question the field most wanted to ask, what does each mark alone do, was exactly the question its own material could not answer. The reagent carried the confound. The glassware was contaminated with the very thing under study. So, around 2014, a quiet race began to build clean tubulin from scratch, recombinant tubulin, one stone model at a time, made where no brain had ever marked it. 3 routes were found. Yeast chimeras, tubulin bodies from yeast, an organism that marks its tubulin almost not at all, with mammalian tails grafted on, a clean body carrying the tail to be tested and nothing else. Baculovirus, a virus put to work in insect cells, brewing fully recombinant dimers, both stones entirely synthetic, with the caveat that the stones arrive wearing a tag to make them purifiable, and a tag is itself a small lie. And tubulin from HeLa cells, a human cell line, as a low-mark middle ground between the soup and the synthesis. No route was perfect. Together they were a new instrument. It is worth pausing on what the race was for. Not a better reagent for its own sake. The entire question of this account, whether marks mean anything, can only be answered by holding the marks one at a time. A field built on the soup had, in a precise sense, never once seen its own subject. Instruments are stories. The story of the soup is that what it taught still stands. Averages are not lies. They are averages, and the preferences and the dial were real enough to show through the blur.
The story of the clean tubulin is that every one of those measurements can now be taken again, one stone model at a time, one mark at a time, and asked to be true not on average, but one by one. The account the soup gave was true in outline. The clean glassware will say where the outline was drawn too smoothly.
There remain the minor marks. They are part of the record, so they belong in this account. But the record here is thin, and it should be read thin. No stories attached to most of them. One breath per mark. Phosphorylation. A phosphate group fastened to serine 172 of beta-tubulin by CDK1, the kinase that runs cell division. Seen in dividing cells, where it may touch the tube's dynamics during the work of splitting one cell into two. What it does is not settled. Polyamination. An enzyme called transglutaminase fastens small amine molecules onto a glutamine, position 15 of beta-tubulin, near the stone's other end. It works on loose stones and on built tubes alike. The bond does not come undone. The mark is for life, probably stabilizing. Little follow-up. A second vinegar mark. Acetylation again, but on beta-tubulin at lysine 252, near the seam where one stone joins the next, placed by an enzyme called SAN, possibly a matter of how readily the stones assemble. Little follow-up. Beyond these two, the acetylation surveys have reported more than 10 further sites, none yet confirmed. Then the reported and forgotten: palmitylation, ubiquitination, glycosylation, arginylation, methylation, sumolation, each seen, each named, each filed, mostly without follow-up. Nothing here should be made pretty. Some of these marks may turn out to matter. The record does not yet say which. The account keeps them the way a ledger keeps small entries, one line each, dated, unreconciled. The landmarks stand because the plane between them is plain.